Okay, so we're here with Professor David Sanders of Purdue University, and David is going to explain to us a little bit about viruses. So, David, take it away. What is a virus, first of all?
So first, I want to discuss what the word virus means. And it comes from the Latin word for poison, virus. But in fact, we have to think about viruses as living things, as biological agents, and they aren't actually designed to cause harm. They are not primarily a poison. They are not primarily harmful. They are just a life form. So we're going to approach the question of what is a virus from a multiplicity of perspectives. So just think about it operationally. It is an OIMP. A virus is an OIMP. What is an OIMP? An obligate, intracellular, molecular parasite. So I'm going to break down all of those words for you. And as Aristotle tells you, a definition both includes and excludes. So let's start with parasite. We all think we know what a parasite is. Parasite is something that lives off something else and doesn't really contribute anything to it. But there are lots of different types of parasites. A tapeworm can be a parasite. A bacterium can be a parasite. So just the word parasite would not be enough to describe what a virus is. So we then move on to what does distinguish it. Well, it is not based upon cells. So the virus itself is not a cell. Organisms such as ourselves are built up out of cells. Bacteria are single-celled organisms. They are cells, but a virus is not a cell.
So why don't you? So just to explain what are the constituents of a cell, and why, and what are missing from viruses that they're not cells.
Right. So we're talking about the definition of a cell. First is a cell will have a cell membrane that encloses its contents. Cells have subcellular compartments that perform particular tasks. They are also capable of self-replication. So they can take in nutrients and convert them into new materials, and they can grow and they can replicate on their own. So a parasite requires a host to grow. So that some cells can be parasites, but a virus is strictly dependent upon a cellular host. And furthermore, what we get out when we reproduce this particular parasite is not a new cell. What we get is new molecules. And so that's the last two words. So it's a molecular parasite. Instead of being an organismal parasite, that is, the product of the parasitic act is a new organism, or a cellular parasite, so a bacterium, we get out new bacteria when we have replication. In this case, the replication of the entity, of the biological entity, just produces new molecules, no new cells.
Got it.
But not only that, then it's an intracellular molecular parasite. That is, it only grows inside a cell. So there are bacteria that tend to only grow within other cells, sometimes bacterial cells, sometimes mammalian cells. But in this case, the virus must be intracellular. That's the only way that it can replicate. And when I say must, that brings us to the last word, obligatory intracellular molecular parasite. There are other intracellular parasites that can either reproduce inside a cell or outside a cell. In case of a virus, it can only reproduce inside a cell, and what we get out are not new cells, but new molecules. So it's an OINC.
I see. So I take it from what we've said so far that viruses do not have their own energy source or ability to use up energy. Is that correct?
That's largely correct. So when we talk about energy source in the, when we're here, they may have a small number of molecules that can provide energy, but what they cannot do on their own is convert energy from the outside into energy for themselves. So they don't have that capacity to use metabolic pathways on their own to create energy. They actually have a store of energetic molecules in them. Many of them do, but they don't have the capacity to create new energy.
And so they're essentially parasitic because they feed they in order to do what they do, and we'll come on to what that is, I'm sure, quite soon. They require the host to provide them with almost everything in terms of their ability to do what they do, including energy to reproduce.
Right. So they need the cell in order to perform many of the tasks that are necessary. I mean, they supply a number of the important elements that are specific to their own replication, but those can't act in the absence of they can't act to really reproduce the whole virus in the absence of a cell.
Okay. So it begs the question then, where on what basis did they come into the evolutionary history of the world, let's say? And,
well, that's
yeah.
a great question, because I actually believe that RNA viruses, coronavirus, for example, Ebola virus, influenza viruses, are RNA viruses. I actually believe that they are reflections of the earliest forms of life. So it seems paradoxical. Now they require a cell to replicate, but I'm also telling you that they could very well be the earliest forms of life. Well, how could they reproduce if there were no cells? And the answer to that question is they reproduced very slowly before there were cells. And now that there are cells, they have learned, and I'm speaking, I know, anthropomorphically, but they have learned how to use cells to obtain the materials and obtain the machinery that they need to replicate more efficiently. So in the past, they would have replicated very slowly on their own, using materials that are present in the abiotic environment and creating some of their own materials. But now, because of the existence of cells, they have, obviously, since the cell provides this machinery and these pre-made materials for them, viruses that use that are going to outcompete any ones that are just trying to replicate themselves in the abiotic environment.
I see. So is it your view also that viruses are older in their form than cellular creatures are?
Yes, yes. As a matter of fact, I see them as a reflection of what other people refer to as the RNA world. And I'm specifically talking about RNA viruses, not DNA viruses. We'll come back to that in a little while. But I do think of them as a reflection of those early. They have evolved over time, but some of the processes that we now think of as cellular probably arose first with viruses or with RNA in the RNA world, RNA-dependent mechanisms. DNA, if you think about it, which we all think about now as being the genetic code and so on, that's a later development in the history of life. RNA comes first, and since RNA viruses don't actually rely on DNA, the ones that we're talking about don't actually rely on DNA, they are likely to be reflections of that earlier form of life, which didn't require DNA at all.
Okay. So So should we talk a little bit about what the most general form of virus is? What are its basic constituent parts? What define a virus? Maybe you can draw. Maybe you can give us a little drawing of what a typical virus might look like.
Well, I don't know if the drawing is helpful at this point. I'll see what I can do. But the most important point is that a virus is a nucleic acid, and that nucleic acid can be RNA, it can be DNA, it can even be a mixture of RNA and DNA with a protein coat around it. So those are the essential constituents of a virus. Now, that's enough to talk about what are called non-enveloped viruses or capsid viruses. So that's sufficient. And that would be, for example, the rhinoviruses or poliovirus. Those, they have nucleic acid and they have a protein shell. There are other viruses, which include viruses such as Ebola, influenza, retroviruses, coronaviruses, that have in addition a membrane around them. Just like the cell has a membrane, these viruses have an additional a membrane around them, and they're called enveloped viruses. And they happen to have in that envelope proteins which are responsible for getting them into the next cell, allowing them to attach and enter the next cell. Sometimes they have other proteins that perform other functions as well in that membrane. So
are they more sophisticated, those ones?
Sophisticated. I mean, they have more parts to them. They're not necessarily. They've just decided to explore a different evolutionary niche from the other ones. So there are viruses differ in size. They differ in size both physically and in terms of their genomes. So one could say, the larger the genome, the more complicated, potentially sophisticated a virus is. But that doesn't necessarily mean that it employs a different strategy, rather not necessarily a better strategy. When we talk about evolution in general, just as an aside, what we're talking about is in a particular environment at a particular time. So we talk about survival of the fittest. It's not some sort of global fitness. It is fitness in a particular time at a particular place. So viruses have explored different times, different environments, different places. And so they've evolved different strategies.
I see. So what is you've mentioned that there's a strand of nucleic acids.
Correct.
Sometimes it's RNA and sometimes it's DNA and sometimes it's a mixture. Let's talk about each of those. What's the difference between them?
Sure. So as I was saying, that RNA is really the primordial molecule. It is really the most important genetic material in the history of life. It is a polymer of ribonucleotides. And what is a ribonucleotide? Well, the ribo part means that it contains a sugar called ribose. So that's called ribose. It is attached through phosphates. So phosphates can make multiple bonds, so they can connect to the ribonucleotide that's in front of them and the one that's after them. So that's the advantage of the phosphate. And then there is what's called a base. Now in RNA, there are 4 bases. They are designated, just for simplicity, by the letters A, G, C, and U. And what's really neat about the fact that it has those different bases is that all the different combinations are where we get all of the great variety in RNA molecules. And they have an inherent ability to do what's called base pairing. So I'm simplifying here, but when we think about base pairing, there are a variety of ways that a base pairing can occur. And within the structures of RNA, we can have different types of interactions. But when we are replicating the DNA, RNA, when we're making a new copy, in general, there is a very simple pattern of base pairing, and that is A's base pair with U's, and C's base pair with G's. So that means if I start off with an RNA molecule that has the sequence A, C, G, U, and there is a what's called a polarity to these RNA molecules. They go from one end to another. The chemistry just allows them to be sort of read one way, one end to another. What we have when we replicate that molecule is another molecule, which is not the mirror image per se. It is a reproduction of the template, and then you get a new model. And that new model, if we have A, C, G, U in one direction, we will in fact have A, G, C, U in the other direction. So the A that we're starting with will pair with a U. The C that we're starting with will pair with a G. The next C will pair with a G. The next G will pair with a C, and the final U will pair with an A. In that case, it looks like it's exactly the same. But we could have a pattern, for example, A, 4 A's, and in the other strand that when we're reproducing it, it would be U, 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 U. But of course, when that one is producing a new one, it will look exactly the same as the original strand. So it's sort of like we have something, we make a mold, and then that we use that mold to make a new thing. It will look like the original entity. And that is the amazing thing about what are called nucleic acids, which are a polymer of nucleotides, and in this case, ribonucleotides. With DNA, DNA, what we have is the same sorts of things, except we have something called deoxyribonucleotides. Deoxyribonucleotides. What's that about? Well, the deoxy tells you that it's missing an oxygen. It's actually missing something called a hydroxyl, which is an oxygen and a hydrogen, but it's missing one. So there are the ribose has 5 carbon atoms, and for example, the carbon atom that's labeled number 3 has a hydroxyl, and that allows it to connect to other riboses through the phosphate. So the 3, it's called 3 prime, but the 3 carbon has a hydroxyl that allows it to connect to other nucleotides, and that's how the polymer is formed. In RNA, there is also a hydroxyl and oxygen atom connected to the second carbon, carbon number 2, called the 2 prime carbon. In DNA, that oxygen does not exist.
And in functional terms, what difference does it make?
DNA is a much more stable molecule than RNA. And it's the forms that it takes in 3 dimensions are far more limited. So I like to say, to be provocative, that DNA is really a boring storage form of genetic material. RNA is the real genetic material. It is as if you were to say that it was computers who created history. If you say that DNA is the important molecule. No, computers are just boring storage entities for the history that we have deposited in there. We created history, we wrote history, and we've deposited it in the DNA. The same thing is true with RNA. We deposit it in the computer. The same thing is true with RNA and DNA. RNA is where the real history of life is. DNA is merely the storage form from which we retrieve the information to create RNA. RNA really does all the work. And because RNA is more dynamic, it can in fact perform chemical functions. DNA really doesn't perform any sort of chemical transformations. RNA can.
So, okay, so let's say you've got a typical virus, an RNA virus.
Yes.
Its constituent parts are a strand of RNA.
It can be one or more strands. One or more strands. Viruses have explored every sort of type of organizations. It can be one or more strands. Some of them can be double-stranded. So actually in the virus particle that's transmitted between cells, there is already a replicated RNA molecule. You actually have a double-stranded RNA.
Okay, and okay, but essentially the strand is encoding genetic material, and the purpose of the virus is essentially to replicate itself.
That's correct.
That's what it does.
I would say that's the purpose of any biological entity, is essentially not to transmit. I like to discuss it this way: to transmit its nucleic acid. That's the purpose of any biological entity, is to transmit its nucleic acid.
I see. So how does it get there? How does an RNA virus do that typically, like in general terms?
So, well, there are several different strategies as it happens. Let's start off with something called a positive strand RNA virus. So what am I talking about that? Well, RNA is not only the genetic material of these viruses, it is also what serves as the message for the production of proteins. So it's a template for production of new nucleic acid, and that therefore new viruses. But it is also RNA can be used to be to encode proteins. Okay, so a positive strand RNA virus, that RNA serves both as the genetic material that is in a virus that's transmitted from one cell to another, and also as what's called the messenger RNA for being turned into the viral proteins once it enters the cell. So in that case, what happens is the virus enters a cell, and its first task, it has no capacity to replicate itself. Its first task is to be translated by the cellular machinery into the viral proteins that are necessary for replication. So it has to find the cellular machinery. That cellular machinery is called ribosomes. Those ribosomes, which is, the word ribosome just means it has ribonucleic acid and it's a body. That's why it's called a ribosome. And those ribosomes, there's actually ribonucleic acid in the ribosome itself, and it interacts with RNA molecules. RNAs do everything in the ribosome. So they are responsible for creating the proteins. They are responsible for bringing the amino acids that are necessary to form the protein polymer to the ribosome. And it is the messenger RNA, which tells the ribosome what amino acids to include in the polyprotein that it is synthesized in the polypeptide in the protein that it's synthesizing. So
Let's just- yeah.
right, the first task of this RNA molecule is to find a ribosome that will will use the message as the basis for creating viral proteins.
Now, are there for any given virus, how specialist are these tools that you're talking about? In other words, will a virus only be able to invade certain cells and be successful in its replication? Or can a virus enter any cell and hijack the cellular architecture to replicate itself? And if the former, if it's only certain cells, how does it how does it get there? How does it know which cells? Or does it just trial and error, or what?
Okay, well, this is pretty far afield, because that is a whole separate topic about the entry of viruses into cells. So they have a whole process of figuring out which cells they're going to enter, but there are other what are called restrictions on virus replication. It turns out that even once it gets into a particular cell, some cells it just can't replicate in. So there are multiple steps in determining whether a virus can replicate in particular cells. And so this can be, there are viruses that have very broad specificity, and then there are ones that have much more limited specificity. And if we think about that specificity, it can be at the host level, the actual animal level. It can be at the organ level. It can be at the cellular level that it has those specificities. So all of those come into play. I did want to just return to the discussion about virus types and virus replication, because I think this really illustrates the idea that viruses are, again, life forms. They are just as variable, just as interesting as any other life form, and they are probably the most abundant life form on the planet. And they can infect any type of cell. So when we talk about the most abundant life form on the planet, we're talking about viruses of bacteria. And there are just enormous numbers of those particles that infect viruses that infect bacteria. So I wanted to just talk about the fact that in this particular case, when we're returned to positive stranded RNA viruses, what they need to do is get the message used in order to make protein. And the first protein that they need to start making is the one that will replicate the RNA molecule. And again, I'm approximating here, but that's what they need to do, because in order to ramp up the infection, they need to make lots and lots of copies of themselves. Independent of producing new virus, what they have to do first is produce lots and lots of new copies of the RNA. So what they make, they come from this. So once we produce that protein, which is frequently called an RNA-dependent RNA polymerase, or the replicase, once they produce that protein, what it has to do is copy the incoming RNA and make a mold from that template. And then what they have to do is make more of that original template, because the original template is what is necessary to make even more protein. So I will illustrate this on the board at this point. So if we have the RNA molecule, which comes in from the virus, and I'll just call it a plus strand so we can understand what we're talking about. That has to interact with ribosomes and make more proteins. Okay, that protein, and we only have one of these. I mean, this is a very hard working RNA molecule. Okay,
Right.
it has to get to a ribosome. It has to be translated into proteins, and then those proteins have to use that molecule. To create more molecules. And what happens is that protein comes along, and it'll start from this end, and it will make, I guess I'll color code this for simplicity, a copy. It'll go in this direction, and this is now called the minus strand. Now, this is not, I mean, these are not identical, as I was trying to explain before. This is a complementary, in the nucleic acid sense, strand, not the same strand. It is a complementary one. But then the protein, again, the protein that we've made, and by the way, I should point out that this molecule that we've started with, the plus strand, can interact with multiple ribosomes, and therefore we can make multiple copies of the protein. We're not just necessarily stuck with one protein. We have one initial RNA molecule, but that RNA molecule can be used to make multiple proteins. All right? So we can have potentially multiple proteins, and that's valuable because we're going to want to make a lot of these. So we're going to be making, from this one thing, we're going to be making a lot of these. And this is the amplification. So we're getting, and that's all what we're talking about in terms of viruses, amplification. Each one of these can then be used by that replicase protein. And sometimes it's a modified, it's slightly modified so that it can work on this one rather than this one as a template. But in any case, it comes along and it then makes, each one of these makes multiple copies of that. And each one of these, again, I'm simplifying here, has the capacity to go back to the ribosomes and make more proteins. And that's how we get this exponential growth of viruses. Go ahead.
Got it. So just to recap so far, we have in any given virus particle, we have the strand of RNA. Let's say if we're talking about an RNA virus,
and we're talking about a positive strand RNA virus.
Positive strand RNA virus, and we have a protein. We have the protein coat.
Correct.
So when it's just sort of floating around in the air, in the atmosphere, that's its structure.
Right. And
what is
that's true for a non-envelope virus, and an envelope virus, it has in addition a membrane around it.
I see. Just talk a little bit about the protein coat.
Sure. That's a fascinating, fantastic subject. And so it turns out that these viral coats tend to be consisting of either one or a small number of molecules. That is, particular one or a small number of proteins that make up that call. And when I say small number, it varies. I mean, but it's not. So it turns out that what happens is that we have in viruses what's called self-assembly. It's largely self-assembly, and we use the same protein over and over and over again to make a coat. So we're using multiple protein molecules, but they all have the same structure, more or less. Simplifying here once again, they more or less have the same structure, and they're used over and over again. And so, and many viruses have different types of what's called symmetry.
Okay.
And so by having these, what's been produced through evolution is you're using one or a very small number of proteins, and they fit together to form a shell that has particular symmetries. And the most common symmetries are called icosahedral. Icosahedron is a 20 faced polygon,
Right.
sort of like a soccer ball.
Yeah.
And they form, and that structure around it. The other type of symmetry, which is fairly common, is called helical symmetry, where they coat the nucleic acid with proteins, and then if you think of a washer, and then you bend the washer slightly so that the two ends don't match up, and you do that with lots of washers, that would give you a helix. And that would be a coating for nucleic acid. So many viruses have those sorts of things. And you can build, the point is that you can build up those coats from very simple constituents.
And what's their function?
Their function is twofold, really: to protect the nucleic acid when it's being transmitted between cells.
Okay.
And to, obviously they have to incorporate the nucleic acid into that structure. They protect, but they interact with another cell, and they actually have to fall apart once they get into that new cell. So they have to both assemble and disassemble. Now, sometimes they may have other functions in, for example, determining where in the cell the nucleic acid actually goes, what's in center of the cell. We don't want it just, floating around hoping to find its right location. Sometimes the capsid can play a role in determining where the nucleic acid is sent.
I see. And so in a typical case of a virus particle invading a cell,
yes,
the protein coat remains outside.
No. So we see that sometimes with bacteriophage, where it seems as if they actually inject the nucleic acid, which in that case is DNA, into the cell. With a typical animal virus, the capsid itself is thought to, at least in part, enter the cell. And that's a complicated issue, because when we're talking about entry into a cell, there are two parts to that. One is going through, getting into the cell, and that is beneath the cell membrane. But it also, another possibility is the release of the nucleic acid into the cytoplasm. So sometimes one can actually get the virus into a cell, but it hasn't crossed any membranes yet. It's just been taken up into the cell without crossing any membranes. And in order for it to replicate, it has to, in fact, cross a membrane to get into a cell. So when we talk about get in, and where the capsid is left, it depends. So it depends upon the virus. It depends whether we mean, is it entering the cell at all, or is it crossing a membrane? All those sorts of different things occur. In the case of enveloped viruses, what we in fact have is a fusion of the viral and cellular membrane. So let's talk about fusion for just a second, and we're going to talk about you for just a moment.
Okay.
And we're going to talk about how did you originate? So that's a question for you. How did you originate?
Well, there's a meeting of genetic material provided by both of each of my parents.
Okay. And the cellular forms those had were?
the cellular forms of, well, of the genetic material?
Yeah.
Egg and sperm.
Egg and sperm, right. So it turns out that the way those two entities become a single cell is through the process of membrane fusion.
Right.
And conceptually, the entry of a virus into cells is very similar to that same process. You have two entities, each with their own nucleic acids, and through the process of membrane fusion, the nucleic acid of one is introduced into the other. So that is, the virus nucleic acid is introduced into the cell through the process of fusion. In those cases, the protein shell is normally transmitted into the cell as well as the nucleic acid.
I see. Okay, so what happens after a little while once the strands of RNA have been copied multiple times within the cell? What happens next?
That's great. So once we have a sufficient number of the RNA molecules that are going to form the genetic material, the viruses start producing the proteins that are going to incorporate those into the shell. So they start producing the proteins that are necessary for making the shell. And then there is
protein coat, effectively.
Yeah, the protein coat. That's right. And so there's no point in producing those proteins really until you've got sufficient nucleic acid to interact with it. So they actually have a timing mechanism, a temporal organization. Again, many viruses. I'm generalizing here, but they will frequently have this temporal organization so that they produce lots of the nucleic acid, and then they will produce the protein, and they will come together, and that will produce new virus particles. And then those viruses have to get out of the cell. And there are multiple mechanisms by which those particles make it out of cells. Some of them leave the cell through a process of fission, membrane fission, which is the opposite of membrane fusion. So membrane fusion, or two membranes come and become one. Fission is the process by which one membrane becomes two. And so that is, again, I'm simplifying, it's a complex process, but so that is one way that viruses can leave cells, just by undergoing a fission event from that cell, membrane fission. Other times,
would that leave the cell intact?
Yes.
Okay.
That would generally leave the cell intact. May not be happy or healthy, but that would leave the cell intact.
Right.
Other types of viruses, including many of the bacteriophages and viruses that do not have envelopes, they are thought to leave the cell through lysing the cell. Lysis. Lysis means really bursting of the cell and release of the virus particles. So what happens there is they make a lot of the RNA, they make a lot of the protein, they make a lot of particles, and then they don't start destroying the cell until they've made lots of them. So once again, there is a temporal mechanism for not lysing the cell, not blowing up the cell, not bursting out of the cell until sufficiently large numbers of virus particles have been created. So those will, in fact, destroy the cell. Now, there is some discussion and some evidence that in some viruses, that even if they don't have a membrane, they can undergo egress. They can be released from cells without destroying them. There is some evidence for that process as well.
Okay, and does the cell sometimes get destroyed just by dint of the sheer scale of the explosion in virus?
So there is the concept. It depends. So there are proteins that are in particular compartments, and when those compartments become overloaded, or when too much of the materials are directed towards those compartments, that can be harmful to the cell. Also, viruses have very clever ways of directing the materials of the cells to their own reproduction and not cellular reproduction. And that can lead to harm for the cell. But also, one of this intrinsic responses of viruses of cells to viruses is to undergo something called programmed cell death, or as it's properly pronounced, apoptosis. And so that is something that cells inherently do. They are willing to sacrifice themselves in order to prevent the transmission of the virus to other cells. So they sense that a virus is reproducing itself. They don't know which type of virus, or they don't know which particular virus. They may know type of virus, but they don't know which virus is there. But they sort of know, okay, I have a virus infection. I am to undergo this programmed cell death. So you might say it is the virus. You might say that it is the virus infection that is promoting the cell death, but also it is the cell death that is a defense against the virus.
I see. Taking the bigger picture, just zooming out a minute and just thinking about what the virus obviously wants. Wants in inverted commas. It wants to replicate itself and to spread itself as far and wide as possible. Is that always and necessarily a damaging thing to the host?
Absolutely not. That is a fantastic question. Most viruses cause little or no harm to their hosts. The ones that we're interested in are the ones that do cause us harm. But in fact, most viruses cause little, they have evolved to cause little or no harm to their hosts. Let me give you an example here. If I were to invite you to my house, you would come in, I'd shut the door behind you, and we'd sit down at the table. There'd be plates and a knife and a fork, and there would be food on the table. And you would be used to being hosted in a home. You would understand the fork and knife. You would see the food and know that it was nutritious for you. And so why would you harm your host, right? I am providing you with all the materials that you need to be, an organism. I'm providing you with everything that you need. I'm providing you with that food.
Right.
So that's true for a virus. A virus, even though, as I said at the beginning, it is the word is derived. Virus is derived from the word poison. That's just how people thought about them, and maybe continue to think about them. I think that that's wrong. They should just be thought of as a life form which wants to replicate. And a life form that's dependent upon its host does not, in fact, from an evolutionary perspective, want to harm its host. All right.
But it is parasitic.
It is parasitic, yes. But that doesn't necessarily mean parasite does not imply harm.
Mm-hmm, mm-hmm.
There's no necessity for harm in that definition. I often tell people that right now it is likely that they have a virus that is unknown to human science. And I often say that to people. And it turns out that just in the last couple of years, we've discovered a whole class of viruses that nobody even knew existed in humans. And it doesn't seem to cause us any harm. And the issue is that much of the harm that comes, and certainly the symptoms that come from virus infections, occurs because of the immune system and the reaction to the virus, not by the virus itself. And we have of the symptoms. Most of the harm that's done by many viruses is in fact due to the immune response. There are three classic sets of symptoms that are associated with the immune response to a virus: fever, inflammation, and fatigue. But all of those are in fact caused by the immune reaction to the virus. Now, I'm not blaming the immune system. The immune system is very important. And if we didn't have the immune system, and that's what happens when people don't have a robust immune system, is that the virus does in fact cause damage and so on and so forth. But most of the symptoms that we actually feel are associated with the immune response. The virus doesn't want to provoke an immune response. It wants to evade an immune response as much as possible. The immune response is generated against the virus. So viruses will evolve not to cause the immune response, not to cause the symptoms and the damage that occur because of the immune response. So the viruses which cause us the most harm, which are the most likely to lead to a pandemic, are ones that in evolutionary terms are recent imports from other animals. Mm-hmm. Or another way of expressing that is viruses to which the population has no previous exposure, right? That's where emerging diseases come from. And so no previous exposure, when we're talking about, so we could be talking, for example, about the exposure of Native Americans in Central and South America to things like measles and smallpox. And that was devastating. It's been estimated that 9/10 of the population was wiped out by the combination of measles and smallpox. So there, what we had is a human population being exposed to another human population that they had not been in contact with, and spread of disease.
Got it.
In the case of these pandemics that we're talking about currently, we have a human population which is being exposed to a virus that is of animal origin. And let's go back to our hosting you.